My name is Michael Scholand. I work for the IEA 4E Smart Sustainability in Lighting and Controls platform, the SSLC platform. And thank you for joining us this morning and this afternoon, excuse me, repeating that. We're going to be discussing the second webinar in our series of three. Today is Circadian Rhythms, Acute, Neurobiological, and Long-Term Effects of solid-state lighting and human health. And today with us, I just want to do some housekeeping. On your Zoom windows, you'll see at the bottom there's a Q&A box. And if you have any questions at any point during the talk, you can click on that and enter your questions that you would like answered at the end, as we will, Jennifer and Sarah will both present, and then we'll have a sort of 15-minute Q&A session at the end. And if you prefer, you can raise your hand, and at the end we will unmute you, and then you'll be able to ask your question live on the air. So so either post a Q&A during the talk, or at the end you can raise your hand and ask a question. So without further ado, let us dive in. I just want to say a couple of words about our speakers this morning. Jennifer Veach is a psychological scientist with expertise in environmental psychology, and she's a research officer at the National Research Council of Canada since 1992. And Sarah is joining us, and Jennifer's joining us from Canada, and Sarah is joining us from Australia. She is currently the Director of Radiation Research and Advice at the Australian Radiation Protection and Nuclear Safety Agency, which is the Australian government's primary authority on radiation protection. So I am going to stop sharing and pass it over to you, Jennifer. Go ahead.
Okay. Okay, so everybody should be able to see that in display mode, I think, if Zoom is working the way it should.
Yes, it looks perfect. Thank you.
Excellent, great. So here we go. I'm going to present the first bit of this presentation, and then Sarah's going to do the presenting in the second part, but I will be driving the slides throughout. So I'm going to start briefly with an explanation of why we're doing this. Apologies if you attended the temporal light modulation session and you've heard this before, but the report that was released a month ago is an update of a report that the former Solid State Lighting Annex had conducted in 2014. And it was time, 10 years, a lot of water under the bridge, a lot of new findings, and also a bit of a shift in general thinking towards a framework of a little more inclusivity, a little more consideration of the needs of sensitive individuals, and an awareness that we need to try to be more inclusive and accessible as well. So it was time for an update. Our goal in the report as a whole was to identify the features of solid state lighting, lamps and luminaires that might be improved to achieve better outcomes. And we wanted to do that both to provide guidance to industry, to, we hope, engage public health authorities a little bit more in this message, and that comes out, I think, quite loud and clearly in this presentation, and also to improve lighting regulations and standards and procurement specifications. So a lot of different goals that we have for generating this knowledge. And being researchers ourselves, we couldn't resist the opportunity to also point out where the gaps are in knowledge, where future research might fill those gaps. It's important to recognize that we had a somewhat limited scope. We were focusing on lamps and luminaires for general interior lighting and street and road lighting. What we were not considering was automotive lighting and light sources that are not lighting products. So no toys, no display screens, no torches that people might carry in case of an emergency. None of that, just general lighting, and only when used in this originally intended purpose. This was a truly international collaboration. As you can tell, we have Sarah in Australia and me in Canada. And we also had collaboration with Christophe Martensons from France. And then other individuals from both Australia and Canada also participated. So it was a really interesting collaboration, lots of meetings between all of us at strange times of day for some of us. I think I never had a meeting at a really awkward time of day. And we were funded generously by the Australian, French, and Canadian governments and from the SSL Annex itself. And what we were reviewing were reviews mostly that had been published in the intervening 10 years since the previous report from the Annex. But where there were no reviews, we looked at individual published research papers and, of course, also incorporated whatever knowledge there is from standards and guidelines and other kinds of information. So we have an outline for this to give you a preview of where we're going. I'm going to be talking about the foundations, and then Sarah will talk about actually all three of the other bullets, and then we'll both come together again for the final Q&A period. Now, I hope what I'm about to talk to you about is somewhat familiar to everybody on this call. We've known for, well, I guess probably close to 70 or 80 years at least, about the cones and the rods in the retina. They're principally responsible for vision. The cones are concentrated in the fovea in the central retina, and they're responsible, excuse me, for the signals that give us detailed vision at high luminance, and they also are responsible for color detection. Rods dominate the periphery of the retina, and they're responsible for image detection at low luminance. But about 25 years ago, the world learned that there is also another class of light-sensitive cell in the retina, which have the very awkward name of intrinsically photoreceptive retinal ganglion cell, which I will henceforth abbreviate as IPRGC. And these cells are located throughout the retina, although there's some evidence that may not be perfectly evenly distributed. There may be some spatial variation. That's a matter of some debate. But certainly there are these cells throughout the retina. And their purpose is not image. Their purpose is simply to detect the presence of light. And that's kind of interesting when you consider that the light has to actually pass all the way through through all these layers to get to the rods and cones, and then the information comes back to the ganglion cells, which are actually on the outer layer of the retina. So it's a pretty complicated structure. Now, what happens with that signal that comes out of the retina? Well, that's the other thing that we've learned in the last 25 years. Whereas previously it was known and thought to be sort of the main event, that the signals that the retina generates would go through the primary optical tract, through various brain structures to the visual cortex, and they're responsible for visual processing. That, of course, still continues to happen. Very important pattern. In fact, you're able to see me now if you have that sequence of cells activating. But what we've learned is that there's also this second pathway through the retinohypothalamic tract, which goes to the suprachiasmatic nucleus of the hypothalamus. It's quite well known and very well characterized that that's how the signal goes through various other structures to get to the pineal gland, where it's responsible for the release of melatonin. That is to say, when light is present, melatonin is suppressed. When light is not present, melatonin is released. And that is really the primary way in which circadian rhythms and the whole sort of timing of physiological processes is cued for the rest of the body. And that's really important. It's also a relatively easy process to study because there are good tests for melatonin in blood and saliva. What we know exists but do not have nearly as solid a handle on the characterization of is how all of these other brain structures get information about the presence of light, because we know there are these connections to the suprachiasmatic nucleus, and we know that that traces back to the IPRGCs, but we don't know all of the different functional relationships of those signals and all of these other brain structures. So there's a lot more still to learn, even though we're in a position of knowing vastly more than we did 25 years ago. it's a pretty exciting time to be in the lighting world. That being said, that means it's all very complicated. We have light influencing everything all at once. Simultaneously in any individual, we have the light that's going through the cones and rods, the cone and rod dominated effects, visual performance, very well studied, visual experience, how we interpret the world around us. Is it beautiful? Is it not? Is there a bright thing over there that we're being called upon to look at? Visual comfort. Is there a glare source? Something so uncomfortable that I want to look away from it? All that's happening in parallel with the exciting new knowledge that there's also these IPRGC influenced effects, some of which are circadian and some of which are acute. And what we mean by circadian is it's happening on a roughly 24-hour cycle. And as I mentioned, that's cued by the presence of light and darkness, the signals that come from the IPRGCs. But it's also a series of effects that are happening acutely. That is, if you're exposed to light, these acute effects happen more or less immediately. And all of those processes on that sort of third layer down in this chart are happening simultaneously and generating the behavior, the physiology, the sense of well-being that we all experience from moment to moment. So although people may talk about they want to do light to generate one kind of effect, there's an unfortunate phrase that's often used, circadian lighting, which many of us just, it's like nails on a blackboard to hear that phrase. It's an unfortunate one, largely or partly because it's really not the whole story. We should be remembering that all of our lighting systems are having to do something to influence all of these processes simultaneously. So we have to understand each of them as individual processes, but then we have to be able to roll them up into a ball to understand the experience of the individual at any one time. Now we have a phrase in the International Lighting Vocabulary to describe this. We call it integrative lighting. It's lighting that integrates both the visual and the non-visual effects and produces physiological and psychological effects on people. And when we talk about this, we do mean this full range of effects that is happening in any one person. And it's exciting, as I say, but also complicated. We also have a system of metrology for these effects. CIE standard SO26, published in 2018, provides SI compliant quantities for assessing light exposures for the full range of physiological and psychological effects. What this standard recommends is that if you are going to be considering effects that include effects involving the IPRGCs, that one should report all 5 photoreceptor types and in terms of the quantities. So the melanopic quantities are those that are reflecting the stimulus to the IPRGCs. The S-conopic, M-conopic, and L-conopic, each of which reflects one or another of the cone cells, and then rhodopic reflects the rod cells. And what this chart is showing, overlaid on all of this, is V lambda in the green curve, which is, of course, the spectral luminous efficiency function, which is the foundation of all photopic photometry. The other important thing to know, however, about what that standard recommends, in addition to using the 5 quantities and reporting them, is that we need to be shifting our focus about where we measure light. Anybody who's been doing lighting design or lighting application for a long time is familiar with going into a location and measuring illuminance, and they usually mean doing that in a horizontal plane. Might be on the ground if it's a roadway situation, or it might be on a desktop or a countertop if it's a work plane. What we need to be thinking about instead of the work plane or whatever horizontal plane is of relevance in the place you're in, is to be talking about a plane at the eye. So we're interested for these kinds of effects in how much light is getting to the eye. And that's the change in focus that I think is possibly going to be among the most challenging parts of this transition to understanding this beautiful, complex problem that we have. And to give you an example of one of these quantities, and how it's calculated. I've chosen to use melanopic irradiance. Melanopic because it's reflective of the spectrum of the iPRGCs, and for most people, that's the photoreceptor they're most excited to learn more about. So imagine for the moment you have a light source. It's an LED. It has a spectral power distribution as shown in the upper left. And you take that and you multiply that irradiance by the action spectrum for the iPRGCs. And the result is you find out that the melanopic irradiance, spectrally expressed, is the chart at the bottom because you've multiplied those two things together. And to come up with the total melanopic irradiance, you would integrate over all of those wavelengths between 380 and 780 nanometers. Now that sounds pretty complicated. Of course, to do that, you need to have a spectral measurement of your light source. There isn't really a simple conversion between photopic illuminance and any of the alpha-opic quantities. So that's a difference as well in the way we measure light. But fortunately, there are several free online calculators that you can use to do the calculation for you once you have your spectral measurements. So it's not quite as scary and complicated as you might think. Now, SO26 also recommends that we talk about equivalent daylight illuminances as a foundation for providing a comparable quantity that allows us to compare between the different alpha-opic values. And the recommendation to use daylight illuminances, which uses the D65 standard illuminant from the CIE as the sort of equivalent, sort of the denominator of the equivalence, is because we know that daylight is so important. So it provides a handy way of tying our conversation to something that we know is important to human experience. And by producing the alpha-opic EDI, as we say, so melanopic or M-conopic or rhodopic, whatever you may choose as your quantity of interest, it expresses the illuminance or the irradiance that you have in terms of the illuminance level of daylight D65 that would produce the same alpha-opic irradiance as your test source. So once you've done that, you have a value that's in lux, and you can compare that to other values. At that point, I'm going to stop, and I'm going to turn over the podium, I guess, or the virtual podium to Sarah, and she will take us through the rest of the presentation.
Thanks, Jennifer. And Jennifer's done all of the abbreviations for me, so I will probably not be defining most of those as we go through, but hopefully if there's any new ones, I won't forget to do that. So if we can just move to the next slide, I'm going to start talking about the circadian and acute effects that we reviewed for the health report. So as Jennifer introduced, the focus of this health report was an update on the previous 2014 report to look at the possible effects that lighting may have on a number of different health endpoints. So one of those is we've termed circadian and acute or short-term effects. So as Jennifer already mentioned, a large part of our physiology and behaviours are really driven by these circadian rhythms or 24-hour rhythms within our bodies. And daily patterns of light exposure are really important, or are the strongest setter of these circadian rhythms. So we know that appropriate light exposure supports not only good quality sleep, but our alertness during the day, as well as many, many aspects of our human health and well-being. Next slide, Jennifer, please. So if we start off with sleep, I think this is one of the most commonly looked at endpoints in terms of lighting, and the relationship between circadian rhythms and sleep is well established. So sleep is really driven by two key processes, a homeostatic process, which is termed process S, and that really is our drive for sleep. So the longer we have been awake, the higher our drive for sleep. And the second process that drives our sleep is process C, or the circadian process. And that runs on a 24-hour clock. So it's kind of like a clock inside of our body telling us it's time to sleep or it's time to wake up. And those two processes interact in order to regulate our sleep. And as I just mentioned, light is the strongest Zeitgeber or timer of this circadian rhythm. So not only does it help to keep it aligned, it can also end up in it being misaligned if we have light exposure at inappropriate times of the day. So inappropriate light exposure can disrupt this circadian rhythm, which then leads to disruptions in sleep onset and quality. And it's potentially connected to other effects such as neuroendocrine, metabolic or physiological effects. However, the evidence for these effects is much less clear than what we know about sleep. So there's definitely more research needed there. Next slide, please, Jennifer. So to give you a little bit extra information about these circadian effects on sleep, so we know that light exposure in the evening can delay sleep and affect our sleep quality, but it's a little bit more complex than that. So if you're exposed to dim light right before being exposed to bright light in the evening, that can actually have a larger impact or further delays to the circadian rhythm and therefore sleep onset. But alternatively, the light exposure that we have during the day can also impact our sleep. So if we have bright light exposure during the day, this can actually reduce the impacts of light in the evening, and at times can actually improve sleep quality, especially if somebody's circadian clock is misaligned. Bright light treatment can actually work to do that. So it's not that easy to understand exactly how light is working, because it's not just the light exposure you're having right now or in the evening. It's also dependent on the light exposure you've had previously across the entire course of a day. In terms of the light itself, we look at light in wavelengths, and we know that shorter wavelength light, so sort of in that blue range area, has a greater effect on our sleep, because it has the largest effect on suppressing melatonin, which is part of that circadian rhythm. So it follows the melanopic action spectrum. And what we know is that someone can actually tolerate a greater quantity of long wavelength light because it doesn't have as large an impact as opposed to short wavelength light in the evening. So it won't have as much of an effect. And that's something that's important to consider when developing lighting, especially lighting in the home. Next slide, please, Jennifer. So in short, we know that bright days and dim nights are the best. And really what that means is the best circumstances we can have for our bodies and our biological clock is something that is really natural. So our bodies are used to having daylight and light during the day and darkness at night. That's what we're adapted to. And so we need to consider that in our indoor and lighting, because we want to make sure we keep it as close to what is biologically normal in our everyday environment. So in terms of how to look at it, we know that melanopic exposure quantities are the best exposure index, as Jennifer mentioned, and the total quantity of exposure is the relevant metric. But the same melanopic quantity can be delivered with less energy if the spectrum is appropriately tailored. So again, there's some complexities here, but there are certainly things that we know now that we can look at when developing lighting. Next slide, please, Jennifer. So if we move on to acute effects, specifically looking at cognition and alertness, when light has been looked at in relation to this, it's the intensity of light exposure that's most widely studied. And self-reports and anecdotal evidence have reported increased alertness with light exposure. However, the effects that are measured physiologically, so when we're not looking at self-report and anecdotal and we're looking at more objective research, the results are inconsistent. So there's less consistency here. And we also know that effects may differ for or between simple and complex tasks. So it depends what we're actually looking at on what we actually might find or see with light. But again, even though some small effects may have been found in these areas, they generally don't translate into what we would say are significant performance differences. Next slide, please, Jennifer. So what we do know, though, is that light exposure during the evening or night can have stronger effects on alertness than those during the day, and that interestingly, seasonal cycles may be implicated in the response to light exposure. And specifically what we mean by that is that greater illuminance or lighting in the morning, but not the afternoon, has been shown to improve alertness, but only during the autumn and winter months. And that's potentially because generally during the autumn and winter months, there is less natural daylight that we are exposed to. And so having that extra lighting that we produce ourselves may be able to have an effect, as opposed to summer, where depending on where you live, you might have quite a lot of light, natural light during the day and sometimes into the evening if you're in Scandinavia. What there isn't any evidence for at this stage is a difference between short intense exposures and long low intensity exposures when looking at this. Next slide, please, Jennifer. So if we then look at well-being, well-being's a little bit of a difficult one to look at because there are many different ways we can define well-being, but generally average daily light exposure is deemed to be too low in industrialized societies. But, and I think as most people probably on the call know, there is still no accepted recommended level when it comes to well-being. So that's obviously something that we need to look at into the future as well. But making that difficult is there's inconsistent evidence for higher light exposure leading to more positive mood. And we need to also consider the balance between visual comfort and visual performance that are required to achieve any benefits that might be there for well-being, such as changes in mood. So I'll move on to the next slide. So when we're looking at well-being, there's only low quality evidence for high CCT light sources improving feelings of vitality and concentration. There's conflicting reports related to them causing less positive mood. A consideration of the spectrum on health impact needs to be balanced against the preferences for colour appearance and high colour rendering. So a little bit like the sleep story, well-being is quite complex. And I think the other thing that we need to take into consideration is that the individual differences in a measure like well-being as well can be quite large. And so it's a really difficult endpoint to get clear indications about. But there is good evidence from the lab and field investigations of light levels and the light distribution when lighting is judged to be good. The individuals tend to experience better mood, better well-being and a work focus. So I think that is something to take into consideration when it comes to well-being. Next slide. And then just overall, morning light exposure has been shown to have a more consistent relationship with improved mood than afternoon exposure. There is some evidence that bright light is tolerated better in the morning and afternoon than the evening. However, there's minimal evidence for the effect of light exposure duration, so how long the light exposure goes for on mood. So there is some emerging evidence coming out there of things we might be able to consider in the future. Next slide. So after looking at those sleep and acute effects, the current expert consensus on light exposure is shown here. So these recommendations came from a meeting of 18 experts that gathered at an international workshop in Manchester in 2019. They reviewed the literature and based their recommendations on numerous papers with data from healthy young adults, looking at a variety of IPRGC related effects, including alertness, suppression of melatonin, that's circadian hormone, and circadian phase shifting. And so what the recommendations are is that during the day, the minimum exposure at the eye should be 250 lux. Before bed, so 3 hours before bed, that should be reduced to a maximum of 10 lux. And then overnight or while sleeping, obviously the best thing that we can do is to reduce this exposure to light dramatically to almost nothing. So to less than 1 lux is the recommendation to ensure that lighting is supporting our health and well-being as much as possible. Next slide, please. However, I did mention when I was pointing out these recommendations that these were based on research with healthy young adults. And as we know, the majority of the population are not healthy young adults. And there's a lot of considerations that need to be thought about. So, for example, shift workers, older adults, young adolescents and children, they all have differing light requirements than the general population. So we need to think about that and how this might change or be different depending on those other populations. But there's also special conditions as well that we need to look at. So people that maybe don't have freedom of movement or don't have control over their lighting conditions so much. So hospital patients, for example, and that's a really important one because we know how important, for example, sleep is in terms of helping healing and all of the rest. And if you've got a hospital patient, but they're exposed to light throughout the day and throughout the night, that's certainly not going to help their recovery from whatever they're in hospital for. But then we also have residents in long-term care, as well as people like prisoners that we need to consider might need slightly different lighting arrangements or currently have different lighting arrangements that maybe we should be considering looking into changing. Next slide, please. So what the report also did was highlight some research gaps, and there's quite a few, and these, I guess we've highlighted some, the non-visible wavelengths and sensitive populations, which I just touched on a little bit. So although these initial results are promising, there's a lot that we still need to learn about the potential uses of longer wavelength radiation, both in the visible range like we've been talking about, but also the non-ionizing radiation region more generally. Also, ultraviolet radiation might have some benefits for well-being, but the difficulty we have there is obviously the substantial risks that come with exposure to UV radiation. So we do know that UV radiation is a carcinogen, and we have to be extremely careful incorporating that into lighting or being exposed to UV radiation. And there's also inadequate information about individual differences in sensitivity that would help us form specific recommendations for subgroups of the population. So as I mentioned, in relation to wellbeing, it's a difficult endpoint because of individual sensitivity, but even beyond that, some of the other populations like younger, older populations or shift workers, we still don't know a lot, and there's a lot more work to be done in that space. Next slide, please. So long-term effects, this is our last section. So I'll just touch on a few of these that are in the report. So eye health or myopia or nearsightedness. So it increases the risk of vision loss through retinal detachment, dense cataract or glaucoma, among other reasons. The incidence is increasing worldwide, but especially in East Asia. And what we do know is that time spent outdoors in childhood is associated with a lower probability of myopia. We also know that short wavelengths, so those blue or violet visible light exposures might be protective. But saying that, there are also treatment devices that use long wavelength exposures. So there's no evidence at the moment that a light source is the cause of this, but it's something that we need to keep looking at to learn more. Next. So we then have age-related macular degeneration, which involves the loss of photoreceptors in the central part of the retina. And this is the main cause of blindness in those over 50 years old in developed countries. So there's a number of risk factors: age, smoking, genetics, other health-related risk factors. And we also do know that long-term exposure to sunlight or very intense short wavelength light might be a factor, but again, the evidence is conflicting here. So again, there's no conclusions possible about electric lighting contributions to AMD. Next slide. And then we have, I guess, probably one of the most hotly debated topics at the moment, which is the disruption in circadian rhythms and the possibility of cancer risk. So the IPRGCs respond to light and darkness and provide the signal that stops and starts melatonin released by the pineal gland. So we already talked about that. And that's what's responsible for shifts in the onset of sleep, as well as changes in the quality of sleep. And all vertebrates release melatonin in darkness, and when light is present, it suppresses melatonin. So we know that disrupted melatonin has been associated with tumor growth, and that acute melatonin suppression by light at night is one factor that's able to do this. And those who work night shifts for many years are also known to have higher cancer risk. And so night shift then meaning that they are having light exposure not at the correct biological time of the day, in a way of putting, they're having light exposure when they should be having darkness, and potentially having darkness when they should be having light exposure. So, but in terms of that higher cancer risk, we also know that it's not only because of circadian dysregulation, and there's many other factors that are really difficult to tease out in these epidemiological type studies, like poor diet, lack of sleep, less exercise, and other occupational hazards that we know are associated with shift work, that make it really difficult to know what cause and effect is. And they're all showing associations, but again, it's not so clear at the moment. But we also know that a strong daily light-dark rhythm reduces cancer risk. And so that does give us a hint that really light is important, and we need to be thinking about this in shift work, and that association that we're seeing with higher cancer risk really may be causal. But again, there's no specific product or system that's implicated in this association. Next slide. So in terms of policy recommendations, thanks, Jennifer. The main recommendations that you'll see in the report is about optimizing the lighting scenarios and individual light exposure. So when we're talking about circadian effects and neurobehavioural effects, I guess what we've emphasised a little bit throughout the talk is a strong light-dark pattern every day ensures the best health. And that light-dark pattern is not just about it being strong, but it being at the correct biological time. So without trying to push our body to shift its biological clock outside of the natural environmental daylight cycle is probably best for our health. Increasing daylight exposure is important, and a high melanopic EDI during the day is also important. Solid state lighting used in the evening and at night should minimize that melanopic EDI for day active people. So again, these recommendations are mostly around what we know about day active people. We still have that gap in shift workers. In terms of long-term effects, myopia, we know that there's some sort of association with time spent outdoors. So I think one measure is that children should spend more time outdoors. In terms of macular degeneration, as a precaution, avoiding repeated direct exposures to high intensity light could definitely be a way forward. And in terms of cancer, personal responsibility for this strong daily light dark rhythm where possible, and reconsidering exterior lighting requirements that might help support this. So the real two key facts are optimizing your lighting scenarios and individual light exposure, but also reducing the intensity of exposures at the wrong time to ensure that health and well-being is maintained. Next slide. But then we come back to, again, these sensitive populations where we've said we know less about, but we do know they have different requirements. So older adults, potentially increasing the amplitude of the light-dark rhythm with increased daytime light levels would be beneficial, whereas children and adolescents focusing on less light in the evening and at night. In terms of shift workers, the work and non-work day schedules together with light and dark exposures need to work in synchrony. But more evidence is needed for how to optimise this, and there are currently no clear recommendations or guidelines for shift workers on how to optimise this constant pushing of the biological clock to be outside the environmental clock timing. Next slide, please. And which is the last slide, the last word being light. So that's what we're talking about here today, is an optical radiation, a flux of photons. It's elementary particles, the photons all the same. So each photon carries its own amount of energy and momentum, which determines its interaction with matter. And that's something that we're talking about here today, and basically how it's interacting not just with matter, but also live living tissues. So we know that photons are scattered and absorbed by living tissues. It produces heat, like all types of electromagnetic radiation, as well as photochemical reactions, including photodetection, which is the physiological basis for vision. And I think the important point here is that these effects are technology agnostic. So it doesn't matter what technology is using them. The effects of light at the different wavelengths, intensities, duration, it doesn't matter what technology it's coming from. The results and impacts on health are the same. So thank you for your attention. And I'm sure Jennifer and I would be happy to answer any questions you might have.
That's excellent. Thank you so much, Sarah and Jennifer. That's very good, very clear presentation. And just to our participants, as noted in the chat, please feel free to post questions in the Q&A now as we enter into the Q&A part of the presentation. Or you can also raise your hand and I will unmute you and you can ask your question live. So we have two questions in the Q&A, which I'll get started with now. So the first one is from Luca Slatner. And Luca asks, have there been any studies done on the effect of lighting on neurotransmitters and or reproductive hormone production? Furthermore, has there been any suspicion of correlation between flickering, discussed yesterday, and depression, anxiety, and hormone production? It's a two-part question there.
Well, I might jump in for the first part and then hand over to the flickering for Jennifer. So in terms of neurotransmitter and reproductive hormone production, there's very little research out there. So in terms of making conclusions or recommendations about that, it's just not possible at this stage. So
okay.
You're on mute, Jennifer.
Oh, Jennifer, you. Yep.
I will add, there is a little bit of research that has looked specifically at serotonin that does suggest that bright light exposure increases tryptophan uptake, which increases serotonin production. And it has been suggested in that work that that does link to some of the work on social behavior and improved mood in people who have subsyndromal seasonal affective disorder. So there's that little tiny bit of work, but Sarah's absolutely right. There's not enough of it to make, huge sweeping conclusions about the population as a whole. To answer the part about temporal light modulation, I have been involved in one study in which we tried to assess anxiety, but it's really difficult because anxiety is one of those processes that isn't as instantaneous as some of the visual perceptions that we were talking about in the other presentation. So because so much of the work on temporal light modulation has brought people into a lab for a series of extremely short exposures, it hasn't been possible to study these slower developing processes like anxiety or really mood states at all, let alone hormone production. So that's another research gap.
Okay, super. Thank you both. I have a second question here from Alex Abanda. The question is, how can lighting designers make better specifications? Should we include daylights in our lighting standard, especially on the indoor settings? What are your thoughts there?
I will happily take that question. In fact, several organizations, including the CIE, have for many years recommended greater use of daylight in interiors for exactly that reason. It also is probably going to be the only practical way to achieve these higher light exposures during the daytime without running afoul of many energy regulations. So we need to get better about daylighting and also sunlight control, because the direct sunlight can be a glare source as well, in order to get close to those, the 250 lux melanopic EDI.
Thank you. Sarah, did you want to add anything? Okay.
No, I think that's
good. Okay. Next question from Dylan Phillips. What are the long-term health effects of excessive exposure to working outside of the typical circadian rhythm, such as for night shift workers, on both physical well-being and on cognitive function?
Well, I think, yeah, one of the long-term effects, or possible long-term effects, is obviously cancer, as we talked about, but in terms of well-being and cognitive function, in terms of long-term, there's certainly clearer acute effects that well-being and cognitive function both decline. But in terms of that's potentially related to, again, whether it's related to lighting, because I guess we're here to talk about lighting, or whether that's related to sleep deprivation or sleep inertia or things like that. It's really hard to tease out. So if we're talking specifically about the long-term effects of lighting, because it's changing the circadian rhythm, I guess it's not really known. Other than the cancer that we mentioned, and yeah, whether there is a long-term consistent effect on well-being and cognitive function, again, I think we don't know.
Jennifer, did you want to add anything on that?
Well, the only thing I would add is looking at the question, which is not just about lighting. There are known long effects of long-term shift work that are adverse. People who work shifts have a higher likelihood of obesity and other cardiovascular health problems. So those are complicated problems. It's not because of the lighting alone. In general, we should probably have fewer 24-hour convenience stores, I think might be part of the answer. 24-hour gyms shouldn't be a thing.
Right. Okay, super. Thank you. Next question is from Timothy Shotbolt. He asks, people who indicate that migraines are a continuing problem and indicate that it is the electric brackets LED office lighting that is the problem, even though they are located adjacent to windows with a lot of daylight. Is the LED the issue or a complex mix?
So there's a lot going on in that. I'll tackle it and then Sarah can leap in. It's hard to say what the cause is of any individual's problems. We do know, this is based on the physics, that if there is temporal light modulation from the LEDs, it's certainly muted by the presence of daylight. The amplitude of the varying thing is going to be sort of swamped by the volume of daylight. So it's unlikely, but not impossible, that the migraine could be triggered by TLM. We do know, however, that some people who experience migraines are also very sensitive simply to the intensity of the light. So it's possible that it is a mix and that part of the problem could be the very intense daylight. Or it could be that, and this is going to sound kind of an odd way to say it, but it could be that the daylight is a glare source. So it's not just the intensity of the light; it's where it is and the difficulty in controlling it could be augmenting or making that a more complicated problem for that individual. So it could be any of those things. And I think it is also true that many people are not very good at attribution of causes. So they may find it difficult themselves to tease apart which is the source of their problem because they don't have the opportunity to do a perfectly controlled experiment on themselves. So I would say that we have to trust to some degree what people believe is the cause of the problem they're experiencing. They have the experience, they may be able to attribute it well, but an expert may look at that and parse the problem a bit differently and narrow down to a more precise solution. So it's hard to come up with a general answer to this question. I hope that's sort of helpful.
Excellent, thank you. Sarah, did you want to add anything, your thoughts on that question?
No, I think Jennifer covered it all, so there's nothing to add there.
Beginning to end, awesome. Well, I'll just give it a couple more seconds for anyone to add, any of the participants to add additional questions. That was our last. Good, we have one more. So Dylan Phillips writes, I have a scenario that I found interesting and wanted your take. Dylan, would you like me to unmute you? And then you can discuss your scenario. Please, okay. Let me do that.
Hello to all of you.
Yep, we can hear you. Go ahead.
The scenario I had was an installation that consisted of a variety of engineers who all had the same take. They were computer-based role. What they had above their workspaces was a UGR less than 16 downlight, but still found that to have more perceived glare than something with twice the flux spread over a much larger surface area. All of them complained of migraines. And I just wanted to take on, I guess, the glare metrics that are used, which is in lighting, and whether they're relevant given the perceived glare of something that was deemed to be glare reduced still had the same adverse effects. Does that make sense?
Sounds like an unusual scenario. I'm having a bit of difficulty getting a clear picture in my mind on it. And I don't know if you're planning on attending tomorrow's session, but I know that Christoph Martin says we'll be talking in more detail about glare.
Sure.
So I don't want to steal his thunder. But I think well, it's a completely separate discussion that we don't have time for today about the merits or lack thereof of the ways we predict discomfort that's associated with glare. But one thing is true, which is none of them were developed with anything like a thought of triggering migraine headaches. So I don't think I would expect UGR to be predictive of migraines. And in fact, I think it's probably a pretty complicated relationship between glare sources and these slower developing problems like migraine. Or
it's certainly like
headache.
it is something that most people consider to go hand in hand, is glare and adverse effects like migraines.
Yes,
so it's
but
interesting that it's not.
Well, but what I'm saying is the quality of our metrics for discomfort is poor. So I wouldn't necessarily expect this.
I just find that interesting.
Yeah. I think it's an interesting question. I think that's actually a bigger question than we can answer here today. But if you're listening tomorrow, ask
Appreciate
Christoph.
you trying all the same. I will.
Yeah. He may even address it to some degree in his presentation. I'm not sure.
Well, wonderful. Thank you, guys.
Yeah.
Super. Thank you, Dylan. We have another question that's come in the Q&A box. So Andrew Murray asks, makes a comment first, that very young people and elderly people have different sleep patterns, where it is common in childcare and aged care to have afternoon naps. Should a different lighting cycle, lux level be considered for these facilities to support the differing sleep patterns?
Well, part of this is, I mean, it's not just a sleep pattern, but it's differing sleep needs. So if we focus on children first, their sleep need, depending on age, can be, anywhere as high as 16-ish hours per day. Whereas older adults or elderly is usually between 7 to 9 hours. The reason that the elderly may take naps during the afternoon is not usually associated with, I guess, sleep need as such, but often with other comorbidities, which is often poor sleep at night. So while I'm not overly familiar whether there's a lot of research in this, when it comes to the elderly, I don't think that we should be changing the lighting patterns to be different from an environmental daylight versus night cycle, because their sleep need is related to what's happening with their health otherwise, usually. So a healthy person or a healthy adult should not need to nap during the day. And with elderly people, yes, it is common, but again, that's usually related to poor sleep at night, which may be due to sleep apnea, lots of other things that are disrupting their sleep during the night. For children, it's a different case because, yes, of course, especially very young children that are needing about 16 hours of sleep, that a lot of that is going to happen during the day. I'm not really aware of any research that has looked into changing the light pattern in this cohort. I think naturally people just tend to, and in childcare settings, they actually just tend to dim the lights when it is nap time. But whether the children would naturally nap otherwise, I'm not sure that that's actually been looked at. But I think given that their sleep need is a lot more and requires sleep during those daylight hours, that dimming light is only going to help facilitate that. So I think it would be a good recommendation based on what we know about sleep and light and how it impacts. So I don't know, Jennifer, if you want to add anything.
No, I think you've covered the whole thing there.
Very interesting question. We have a new question from Andrew Gale. So yeah, thank you, Sarah, for that. Andrew asks, there is a focus on CCT and street lighting pushing for 3,000 Kelvin over 4,000 Kelvin. Brisbane City Council and Energex engaged QUT, I guess that's
Queensland University of Technology.
Queensland University of Technology.
Got it. Sorry about this. Okay, Brisbane City Council and Energex engaged QUT to do a study to show there is minimal effect from blue light from street lights, in particular minor road lighting. The concern is the minimum horizontal luminance can be as low as 0.07 lux. Considering our eyes are in a mesopic state, which makes it more difficult to see the road verge at lower CCT. Can you provide advice on this?
I'm not sure I understand the question entirely, because I wouldn't expect, well, first, I wouldn't expect much difference in visual performance between 3,000 and 4,000 Kelvin, regardless of which correlated color temperature you have. 0.07 lux is very low. So I'm surprised that that would be your roadway standard, frankly.
I just unmuted Andrew to ask the question live. Go ahead, Andrew. We can't hear you at the moment. You're unmuted, but we're not hearing your voice. Yeah, sorry, Andrew. Maybe you can check the settings on your microphone. Give you another second.
So while he tries to sort himself out, I'll add that CCT is not a terribly good metric, really, of anything, but it is likely that the 3,000 Kelvin choice has relatively less short wavelength light. And so one would expect a lower melanopic EDI from that light source than the 4,000 Kelvin one for the same scotopic illuminance. From that perspective, that's probably a good thing for the people who live nearby, always providing that the street lighting isn't shining directly into someone's bedroom overnight. As to whether that's appropriate for the roadway from visual needs, I think that's a little bit beyond our scope today.
Yeah, I'll be in the standards.
Yeah. And I note the comment
from Alex.
Yeah.
Yeah. The street lighting CCT change is mainly due to, go ahead.
And that's actually, we had at one point hoped we could cover some of the environmental issues in our report. But that's a huge area of active research these days. It was beyond our capability to cope with.
Right.
And yes, that's exactly, anonymous attendee, that's exactly the concern. It's yes, it's important, and it's also a huge topic that someone else is going to have to tackle.
Yeah, we're all earthlings, right?
That's right.
Yeah. Okay, Andrew, do you want to, I don't know if you want to try and clarify your question in the chat, if that, since your microphone doesn't seem to be working at the moment. And while you're doing that, if there's any other questions, we are at top of the hour, so yeah, actually right on time, I would say. Just give Andrew another second here. Any other questions that folks can think of for Sarah and Jennifer? It's hard to know if Andrew's typing at the moment and how quickly he can type.
Yes.
You know,
Yes.
so
It's
I'm
not like
really.
Teams where you can see the little bouncing ball.
You can see the little dots going, exactly. So they're entering the data. But Andrew, please post your question, or we'll wind things up at this point, and maybe you've got Sarah and Jennifer's email from the slides. Of course, you've got mine. Okay. Let me do that. So Ron Dinesh, let me unmute you.
Oh, but Andrew says he'll send an email.
Okay. I was curious. It's not. I can see he's trying as well. His muting and unmuting, but it's not. We're not hearing anything. Yeah. Okay. Last shot. Yeah. Sorry, Ron. We're not hearing you either. Okay. We will pursue the email path on this. And I would use that opportunity now to wrap things up and say thank you again to our speakers, to Jennifer and Sarah, for taking the time today to go over this section of the health study. And thanks to all our participants for joining us. It's fantastic to have you. And we will be here again tomorrow at the same time if you'd like to join us for the third and final round. And yeah, and a final in this series, I say this round. And again, thanks to everyone, and good day, good evening, and good night. Bye for now.
Bye-bye.